Bismillahirrahmanirrahim. 5090 O-Level Biology, June 15, paper 1-2, question 1 to question 20, and this is the first video, and I would of course like to read this very important every positive change in your life begins with a clear, unequivocal decision that you're going to either do something or stop doing something. Now, it's a very thought-provoking quotation which I would like you to think about when you all walk through life. Start with question number 1. The diagram shows a plant cell. Now let's look at the labels. Which structure identifies this as a plant cell rather than an animal cell? Now an animal cell will also have the cytoplasm. Animal cell will also have a cell membrane. It's difficult to underline the cell membrane, but it will be the inner membrane. I might not be able to do it with this sort of pen. So the inner membrane, there is a cell membrane which is also present in both, and then the nucleus is present in both, and then we have the chloroplast. So now just revising this for you all, which structure identified this as a plant cell rather than an animal? So if you said nucleus, but nucleus is present in both, cytoplasm is present in both, cell membrane is present in both. Only cell wall is present in a plant cell which is not present in an animal cell. Then coming to question number 2, which processes are responsible for the uptake of ions from the soil by a plant and the uptake of glucose into the villi of a human? This is actually the syllabus point. If you look at it, these are the two examples in this chapter which the syllabus expects you to know. So the uptake of ions is by active transport, and these are actually examples of active transport. So it's exactly the syllabus point. That's why I keep on insisting that you must look at the syllabus before the exams. You must go through it very thoroughly. They are going to ask you those particular points. Question number 3. The diagram shows 3 plant cells, P, Q, and R. So we have P, we have Q, and we have R. The arrows show the direction of water movement by osmosis. So what is happening? Let's look at the arrows. It's going from P to R, and it's also going from Q to R, and it's going from Q to P. So that means which one has a higher water potential? So Q has a higher water potential than P. Q has a higher water potential than R, even, because movement is from Q to R and Q to P. And then P has a lower water potential than Q, but a higher water potential than R. Please understand this. What is the correct order of water potential in the cells? Naturally, from Q it's going to P and Q to R. So Q has the highest, and then P, and then R. So Q, P, and R. Highest water potential, so the highest to the lowest. Then coming on to 4th, a dish filled with agar jelly containing starch. 4 holes are cut in the jelly, and each hole is filled as shown. After 30 minutes, which hole will be surrounded by the largest area without starch? Why? Because if it contains an enzyme amylase, so there is an amylase solution, then this is amylase solution with hydrochloric acid, then this is gastric protease solution, and this is the agar jelly. And what did the agar jelly contain? What is this agar jelly contain? What did it say in the question? Dish is filled with agar jelly containing starch. This is what you should have immediately written on the paper. Please remember, you can write on the paper, and that is what you should do, so that you know what you are answering. So the proteases were total rubbish, because the agar jelly contains starch. So it's only the enzyme amylase which digests starch. So here we had two choices. It was either A or B, but amylase in hydrochloric acid, amylase, even whether it's the salivary amylase or the pancreatic amylase, only works in a neutral medium. There is no amylase in the stomach, which is where the HCl is being produced. Now, if I had changed this question and I said dish is filled with agar jelly containing protein, then what would have been the answer? The answer would have been then this. Then D would have been the answer. If I had said this is filled with protein and then we punched holes and we've added something to it. And what did we add to it? Now that is going to diffuse out and is going to digest the protein outside. So understand, now I've made this into two questions. So please understand it, how this question can be changed and how you should know the answer to it. Now, question 5, this has been mentioned in the exam report as well. So this was a tough question and many students got this wrong because of a concept which they were not clear about. Compared with the rest of the cell, which row describes the concentrations of oxygen and magnesium inside structure P? Now, what was structure P? It was a chloroplast. So if the chloroplast, what is present inside the chloroplast is the chlorophyll, the pigment. So there is a pigment called chlorophyll, and what is going to happen here? There's going to be photosynthesis. And if there's going to be photosynthesis during daytime, then there's going to be a lot of oxygen being produced. So high levels of oxygen and magnesium is there all the time because the chlorophyll molecule has magnesium in it. Just like you say for hemoglobin we need iron. So for the chlorophyll molecule you need magnesium. So magnesium तो है ही उसमें, there is magnesium in it. So chlorophyll में magnesium था and it's in the chloroplast. Chlorophyll is a pigment which contains magnesium and is present inside the chloroplast during the daytime. It will photosynthesize and there'll be lots and lots of oxygen. So there was high levels of oxygen and high levels of magnesium. Just confusing you all. Otherwise, you all, I'm sure, knew this. That are green in the center but white around the edges. So it is green in the center. This part is green. This part is green. This part is green. And this part is green. But it is white in the edges. Around the edges, it's white. I haven't colored that any color, but I can color that yellow. Just no, I won't color it any color because then you'll get confused. The plant is de-starched means we've kept this plant. It's a potted plant and we kept it in the dark and then we've left it for 48 hours. And then has one of its leaves partly covered with black paper. So here we have added the black paper, right? So this is the black paper we've added here. So we've covered it, so no light can reach it. So the part underneath it will not contain any starch because that did not receive light. Light is necessary for photosynthesis on both sides of the leaf. So we placed it on both sides of the leaf, upper side and the lower side. And then what we do, the plant is placed in bright light for 48 hours. The leaf is then tested for starch. What diagram shows the colors? Naturally, the part which was green will be containing the starch, so it will be black. And the part which was white will be brown. Why? Because iodine colors brown when there's no starch present. Iodine is brown in color. If you add it to water, it will remain brown. But if you add it to a starch solution, it will turn black, brown to blue-black. So the others are all rubbish because you see you had covered it. So there would be a patch. Now they look, this is now confusing. This is a lot of students would have marked C. Why? Because they didn't know it. Black is only when starch is present. Brown is when there's no starch. If you're not clear on this, well, you can't do these questions. Black is when starch is present. Now, as you can see in this question number 7, the diagram shows cells in a section through a leaf of a typical green plant and no cell contents are shown. So 1 is the upper epidermis. 2 is the palisade mesophyll, right? And 3 is the lower epidermis. And 4 is the guard cells. Now, it says which cells usually contain chloroplast. Now, remember, it's 4 is the guard cells which usually contain chloroplast. And the spongy mesophyll contain chloroplasts. But they didn't mark the spongy mesophyll. And the palisade mesophyll contain chloroplasts. So, a quick revision. It had to be 2 and 4. 2 is the palisade mesophyll and 4 is the guard cell. 3 is the lower epidermis. That doesn't contain, the epidermis cells do not contain any chloroplast. It's a confusion. If you have that confusion, well, you won't be able to do this question. Then question 8 is again also mentioned in the examiner's report. What is an example of assimilation? What is assimilation? Assimilation means when once you've eaten, say you've eaten roti or you've had rice and you've had chicken kadai, and that is digested and is absorbed, then what happens to it in the cells? That is assimilation. What is the use of it? What happens to the use of the nutrients that you have eaten? Like if you had rice, that rice contains starch, finally is going to be converted to glucose. But what happens to this glucose? This glucose enters your cell where it is respired to release energy. If you had chicken, well, there'll be proteins. The proteins will be digested to amino acids. The proteins will be digested to amino acids. And those amino acids will be used by your body cell to make pepsin, to make insulin. Not all hormones are proteins, please remember that. So, of course, this was all wrong. Absorption of into lacteal, this was wrong. Alcohol, this is not assimilation, it's absorption. Glycerol will enter the lacteals, this is correct, but it's not assimilation. Breakdown of alcohol in the liver, no, that is not assimilation. Then release of hormone from a gland is not assimilation. So basically it was C, which was the building of proteins from amino acids. Then question 9, what are the basic units from which glycogen is synthesized? Glucose. Amino acids are used to make proteins. Fatty acids are used to make fats or lipids. And glycerol combines with the fatty acids. Glycerol and fatty acids combine and fats are made. So if you knew it, well, you knew it. If there was no sort of trick question in this. Either you knew it or you didn't know it. And question 10, a food is known to contain protein but no reducing sugar. When this food is tested with biunit solution, P protein, P present. No, it was P present is 3 P's. Turns purple or lilac or violet. Same thing. It means P purple, P protein, P present. Pakistan People's Party. So lilac, so these two were wrong. We said protein was present. So this would be correct. And Benedict's would be negative, so it would remain blue. It's only brick red if reducing sugars are present. It's only brick red if reducing sugars are present. Then coming to question number 11, the diagram shows transverse section of a plant stem and of a plant root. Now this is something which you all must know. There's no ambiguity about it. Stem, the xylem is on the inside and the phloem is on the outside. Similarly here, the xylem is on the inside and these outer thingies, which I'm just going to color in purple, this is the phloem. This is something which you all must know. And here the phloem is on the outside, so that the aphids can take the sugary syrup. You have to remember it. You have to do some story to remember it. And the xylem is on the inside. So this is what you need to understand is that the phloem is on the outside. So you had to know it. So it was which regions contain the xylem is 2 and 4. Now coming to question number 12. In an experiment to investigate transpiration, the leaves of 4 identical shoots are treated as follows. Upper surface covered with waterproof jelly. So this is, we have a leaf and we cover the upper surface with waterproof jelly. Then this is one. Then in number 2, we have the same leaf. We take another leaf and we cover the lower surface. So the lower surface is covered with waterproof jelly. Then in 3, both the upper and the lower surface are covered with waterproof jelly. And 4th is untreated, just the leaf. And then what did they measure? The graph shows the water lost by the 4 shoots. Now, whenever there's a graph, now there's a way of doing this question. I know this question is not going to come, but a similar question is going to come in your exam. So time, say you write here 1 minute, 2 minute, 3 minute, 4 minute, 5 minute, 6 minute. Just do something like that. Or maybe you can say 5, 10, 15, 20, 25 minutes. And you just imagine that it is a minute. So to say this is now 20 minutes. And amount of water loss, say you say here 1, 2, 3, 4, and 5. And this is in milliliter because it's water. So now where was the most water was lost in A? Then it was very quickly in say 5 minutes, the water lost was 4. Now in the other one in 10 minutes, now let's try to go here. The water loss was only 4. So please remember, if you ever do get a graph question, do this exercise. It is very important that you do this exercise to explain this question to yourself. You're not going to have time, but you have to do it very fast. Now look at C. Now C had less water lost. D had even lesser water lost. C, now I'm going to give different colors so you all can see. So C was this, and then B was this, and then A was this. So A lost the most water. So it says which line shows the results for shoot 4? You see, when you cover the upper surface, some little water will not be lost because there is cuticular transpiration. Lower surface has all the stomata, so of course there will be no water loss. Upper and lower surface both covered, so of course it will be. So if you look at A, B, C, and D, now this was 4. Now this would be 1. This would be 3. No, this would be 2, sorry. And this would be 3. Least amount of water loss will be when it will be both. So now if they turn this question into this question which I've just given you, then I'm sure you would not figure out the answer because if you hadn't done this whole exercise. So please understand, this is very important that you understand these questions. The diagram represents the heart and some major blood vessels, head, lungs, rest of the body. What are the possible blood pressures in kPa for the vessels shown on the diagram? So now let's look at which are the vessels. Now what is P? Now if you look at vessel P, it is coming out of here and it is going here to the body. So what is this? This is the aorta. Then what is S? Coming out of the lungs and going to the left side. Pulmonary vein. What is R? Coming here, going to the lungs, pulmonary artery. And what is this one coming here, head, and coming here to this, and also coming Q, vena cava. Now, what are the blood pressures in kPa for the vessels shown in the diagram? So, the highest pressure would be in the aorta. Because why the aorta has the highest pressure? The reason is because it's arising from the left ventricle. And the left ventricle wall is very thick, so it exerts a lot of force. Aorta by itself can't do anything. It is the left ventricle which exerts this force. So what are the possible pressures? So it had to be 16 with the aorta. Now that was, of course, you had the choice between C and D. There was the only difference. Now what was Q? Q was the vena cava. And R was the pulmonary artery. So pulmonary artery is arising. Arteries usually carry at a high pressure. So pulmonary artery would be more than the vena cava. And then, of course, pulmonary vein would be the least because it's coming from the lungs back to the heart. So this is something which you have to understand, these circulations and the pressure, that the left ventricle had to have the highest pressure, and then of course the pulmonary artery would have a subsequently high pressure because it was being pumped by the right ventricle. So it would of course have some pressure. The other ones are veins. The vena cava would have, you see, you have to understand the vena cavas would have, as compared to S, what is S? S is the pulmonary vein. So vena cavas would have two pressure, and the S, which is the pulmonary vein, would have one. Pulmonary vein is the blood has gone to the lungs and then it's just returning back to the heart, and so low pressure circulation as it is. So very good question, very difficult question. 14, the diagram shows changes in pressure in the aorta, left ventricle, complete heartbeat. What causes the increase in pressure between points X and Y? Now here I've always told you to follow this aortic pressure. This is the biggest clue that you get. So aortic valve is open, blood is flowing into so it is the contraction of the left ventricle which is forcing blood, and that is causing the increase in pressure because the left ventricle is contracting to force the blood into the aorta. So that was the point that you had to really understand, that the aortic pressure in the aorta. This is the graph which helps you the most if you can figure this out. Then coming to question number 15, the diagram shows the liver and its blood supply. What is the name of blood vessel X? Now everybody knows that's hepatic portal vein, which joins the liver to the alimentary canal, or from the intestines to the liver. But then it said the diagram shows liver and its blood supply. What is the name of blood vessel X? Which substance increases in concentration between vessel W and vessel X? So vessel W, it would be carbon dioxide. And how do you figure that out? See, what you had to understand was that the hepatic portal vein carries deoxygenated blood because in the intestines all the oxygen has been used up. So this would be carrying deoxygenated blood. And what would be W? W would be the aorta, because this is the vessel which is going to the alimentary canal. So it's an artery, and this is the vessel going to the liver. So this is another artery. So arteries carry more oxygen, less carbon dioxide. So this would be the hepatic artery. And this would be an artery. So this would be actually the aorta, which is carrying oxygenated blood. And X would be the hepatic portal vein. And hepatic portal vein, always veins usually carry deoxygenated blood. So this is something which you all were not very clear. It's again mentioned in the exam report. Candidates were able to identify the hepatic portal vein, but some did not know that this vessel carries deoxygenated blood. Question 16. Which word equation shows anaerobic respiration and used very simple glucose converted to ethanol and carbon dioxide? Yeast is used to make alcohol. So ethanol and carbon dioxide, and it's also used in bread. We use the carbon dioxide to make the dough rise, and that's anaerobic respiration. Then coming on to question 17. In question 17, the table shows the effect of exercise on the rate and depth of breathing. You can see the working that I've done. Breathing rate is breaths per minute. So 12 into 500 is 6,000. And 24 into 1,000 is 24,000. So what is the increase in volume? You had 5 rupees, now you have 20 rupees. What is the increase? 15 rupees. You had 5 rupees, now you have 20 rupees. What is the increase? 15 rupees. 20 minus 5. So 24,000 minus 6,000 is 18,000. So what is the increase in the volume of exchange per minute? This is breaths per minute. This is volume of each breath. So this was simple that it was a little bit of maths, but some of the students found this very challenging. I still have students asking me this question. Question 18. The diagram shows a section of an alveolus and a capillary in a lung, airflow in, out, blood flow. So they're showing you the blood flowing in and they're showing you the blood flowing out. And says what are the relative concentration of carbon dioxide at X, Y, and Z. Now X, you know, is there's carbon dioxide is going to be very high at X. Why? Because it is deoxygenated flowing into the lungs. So here we have deoxygenated blood. So carbon dioxide is going to be the highest in X. And then Y it is slowly because here the carbon dioxide is going to come out here, but then it's going to be exhaled out also. And then at Z is the blood, which is now mostly deoxygenated, so it should be the least. So that is why the answer was B. The diagram presents a kidney machine. So there's a pump, there's a dialysis fluid in, dialysis fluid out. So the dialysis fluid in comes here, and the dialysis fluid is out of here. And here we have the blood which is going in here. The blood is inside these vessels. I'm coloring them in blue. And these sort of tubes which I've got, and the blood is coming from the patient's arm and then it's going into this machine. Which two substances are both present in the dialysis fluid entering the machine? What do we not want to move? We don't want glucose. So glucose and salts are already present because what is the machine doing? Doing the work, machine is doing the work of the kidney. So we only want the urea to be removed and excess salts to be removed. So what two substances are present in the dialysis fluid? So the dialysis fluid is on the other side, which I'm going to color in yellow. So this is the dialysis fluid all around these tubes, all around the blood. So the dialysis fluid is going to contain what? Which two substances are both present entering the machine? Because then you see the dialysis fluid is going in here but out here. So we don't want any of the glucose in the blood to come out. So there has to be glucose in it. So there is no net movement of glucose. Similarly, there is certain number of salts that we need in the body. We want only the excess salts to come out. So we don't want the salts to be lost in the dialysis fluid. The salts coming out of the blood entering the dialysis fluid, there should be no diffusion gradient. So glucose and salts are going to be present. The rest is all, wrong. Proteins, there's no possibility of any protein presence. So both these are totally wrong. Urea, of course, we don't want, there's no urea in the dialysis fluid. The dialysis fluid is so that the urea in the blood diffuses out and enters the dialysis fluid and is removed from the body because the kidneys are not working. Then coming to the last question, the diagram shows some of the structures in the human skin. Which labels describe the structure Y and Z in hot conditions? Now, Y is the sweat gland, so it's going to be very active because it's going to be producing a lot of sweat. So Y is the sweat gland. It's going to be very active. So it could only have been A and B because it said in hot conditions. You enter this room, it's so very hot. Well, he's going to start sweating. And then Z is the capillaries. So they will be dilated. Why? Because in hot conditions, it's vasodilation. So Z will be dilated. So that's why the answer was B. Okay, so now this finishes this video here, and we will continue the rest of the questions in the next video. Thank you very much and best of luck.